I'm one of the organizers of Neuromatch Academy, and I'm here today to host the question and answer session for Causality Day. I have with me Manjari Nayaran and Adeel Razi and our own Konrad Kording. But first, I would like you to spend a few moments filling in your daily reflections. You know, these are very important for us. We value your feedback. We hope we can use it to understand what we've done well and what we've done badly. So please take a moment, click on the URLs on your screen and fill in your daily reflections. So while you're doing that, let me make a couple of quick reminders. First, please, when you go to the chat, if you'd like to chat with both us and the rest of the students, please select all panelists and attendees. Otherwise, only we will get to see and hear your interesting thoughts. And please ask your questions in the question and answer panel, and we will start taking questions in a few minutes. In the meantime, let me ask my guests to introduce themselves. So why don't we start with Adeel?
Yeah, thank you. Thanks a lot, Eric. So I'm electrical engineer by training. I did until my PhD, I was doing wireless communication, developing ways that how one can transfer information wirelessly from one place to another. And that's until 2012. Then I thought about moving into life sciences and use my engineering skills to model how the brain processes work and how the information is transferred or information is processed in the brain. And I moved into neurosciences 6, 7, 8 years ago now. And then I started at UCL working with Carl Friston, developing what we call dynamic causal models. And then 2 years ago, I worked with him about 5 years, actually learned a lot about neurosciences at UCL, and then moved, so I usually say that I'm like a grad student because I started neurosciences like 7, 8 years ago. So, or maybe a grad student plus 1 year of postdoc. So I don't consider myself as a senior faculty. And then 2 years ago, I moved back to Australia from UK, where I'm now heading a group in computational neuroscience. So I'm based at Monash University at Turner Institute for Brain and Mental Health. And I continue to develop neuroimaging methods with a focus on brain connectivity. And very specifically, I work on resting state functional MRI.
Thank you. And next, Manjari, would you introduce yourself?
Hello, everyone. So I'm Manjari Nayaran. I'm a postdoc at Stanford School of Medicine. My background, actually, I started off in doing signal processing and electrical engineering during grad school, and then I moved into statistics, where I became very interested in inferring, graphical models of complex systems and how we can infer these in finite sample sizes and so forth. I became interested in human neuroimaging during grad school, and during my postdoc in the last few years, I've been focused on better statistical estimation of functional brain networks, what happens when we want to study changes in functional brain networks and how do we do that effectively. And in that context, I've developed a huge interest in how do we really make sure that our dependencies between, for example, brain regions actually reflect causation. And on a related note, very interested in issues of causality in the context of clinical applications. So for example, how do we know that a biomarker that's based on brain imaging to reflect some kind of clinical outcome, how do we know that that's actionable? Like if we find a correlate of the brain and we want to say we want to intervene or do manipulations or develop a new therapy that affects that circuit, how do we know that that's actually going to have an effect on some clinical outcome? So I've become more interested in how to validate biomarkers from a causal inference perspective. And so that's a little bit about myself. Back to you.
Conrad, would you introduce yourself briefly?
Yes, great. I run a lab at the University of Pennsylvania. We've always been interested in how to analyze neural data, but over the last 4 years or so, my lab has almost entirely switched from a mode where we just analyze data to a mode where we're interested in figuring out about causality, because for me, causality kind of maps on the kind of questions that I really want to ask. And I think there's both medical applications, but there's also like, if you ask me what it means for me to have a model of the brain that I want to have, then like causality figures very centrally to that. And I just want to share like one little bit here. It's 2:10 or so here. I haven't been awake at this point of time, possibly in years. And I haven't gotten any chance to have coffee. So I hope that I'll be able to survive through the session today.
Well, it's always interesting to be on these Neuromatch panels because we have people scattered across the globe. I'm in Lisbon, Portugal. I think, Manjari, you're in Stanford in the Pacific time. Conrad's on the East Coast, and we have somebody in Australia. I feel a little out of place here because I don't have an electrical engineering background, and I've just realized that they outnumber us, Conrad, or at least they outnumber you as the panelist. I'll just briefly mention that my own background with regards to causality is my misspent youth as a philosopher, where one of the fundamental questions that we wrestle with is little things like free will. But if you want to think about something like free will, you really have to think about what is the underlying causality that would allow you as an individual, as an agent, to be able to affect things in the world. To do that, you have to have causal action. You have to be able to know that your thoughts and beliefs hopefully cause stuff. And turns out that's a really thorny, hard problem. And philosophers have been struggling with how to deal with this for a long time, especially because we also want our will to be free. And the more you box yourself into a world that is causal, the harder it is to have any freedom. So I spent a lot of time thinking about this back before I was doing neuroscience. So I'll try to interject useless thoughts from that perspective. I also have played a little bit with some of the other tools you guys, I think, have been working with today. But let us begin. So I guess while we're waiting for a few more questions to come up, I think fundamentally, one of the things that might be nice to ask is it really the case that causality is something we need to really care about? Because after all, 99.9% of what we do in neuroscience is just correlations. Isn't that fine? Aren't we good enough? Someone want to just give us a little starting point as to why this day might really actually be interesting and important for us? Everybody seems amused with this question. So.
I mean, like I can, of course, I could of course start. You know, like, what's the question? What's the kind of question that we often want to ask? And we can see what the questions are that we want to ask by looking at our own papers. You know, like we write things in papers like area A is a good target for interventions if I want to have a treatment that affects area B, because area A affects area B. Now, if areas A and B are correlated, that doesn't mean at all that now I could affect area A if I want to have an effect on B. No, like let me give you an example. With TMS, we can only, of course, affect areas that are on the surface of our brain. If I want to change the activity in a brain area that is not at the surface, I would want to have a model that tells me what would happen if I would stimulate at a given place. Now that's a causal question. It could be that that two areas, maybe a deep area and a superficial area, are very, very strongly correlated, but that in no way means that a stimulation of that superficial area would actually make the deep area go up in activity. No? And so the real question that we want to have there from a treatment perspective is we want to have causality. But it's the same thing, the kind of stories that we tell about brains, they're all causal. Not like this neuron makes this neuron fire, which makes this neuron fire, which makes behavior happen. That's the space of models that we have in much of neuroscience. And when I say space of models, I'm talking about like the how models, where we kind of ask how, like, what the mechanisms are that give rise to that. So deep in the definition of mechanism is embedded a notion of causality. And that's why kind of like, yeah, why at least for me, it's not even a question really if we need to get at causality. That is really what implicitly almost all of neuroscience is about.
Yeah, and I think maybe the interesting thing is that it's what we all would like to be able to say at the end of a study. You believe that we know that this piece of the brain causes your arm to raise up, or this piece of the brain is causally involved in, you know, your sense of smell. But very rarely do we even directly test it using the kinds of statistical tools that our guests are you know, working on. So I don't know, Manjari, do you want to add anything? I'm curious whether, you know, what's your first take on why we should care about this?
So, you know, let me give an example of some questions that come up in my own work. First of all, I love the example Conrad gave about TMS and brain stimulation. I have an even more basic question, which is that sometimes we attribute, you know, that we find a phenomenon that, you know, we find an fMRI marker of X, and we presume that that tells us something that, oh, we found something in the brain, something about the neurobiology that's related to some outcome. But it can turn out that that there's something that's completely non-neurobiological that's also like predicting the outcome. So at a very basic level, you know, causation doesn't even need to mean like neuronal level mechanisms, which is what, you know, when I bring up causality, that's often what people think I'm talking about, that I'm, you know, and the neuronal level biological mechanisms are of course extremely interesting, but even more basic than that, I think a lot of biomedical science today wants defined biological mechanisms. So for example, people who study gene expression and how gene expression changes over a lifetime, they want to find how, you know, methylation in the DNA is going to predict all sorts of outcomes. So for example, people found results that said, hey, we can predict aging and longevity through studying chain variation and methylation of DNA. Well, it turns out that even though people think they're measuring methylation of DNA, it turns out that when you measure methylation in DNA, it's based on how genes are being expressed in different cells. And if one person has a certain proportion of cells and another person has a different proportion of cells, then because of the fact that the proportion of cells are different, you find that we can predict age. But if it turns out that, you know, people say we've found epigenetic mechanisms that predict longevity or aging, but it turns out that it's not the methylation, it's the proportion of cells that are doing the work, then have you really found, you know, guaranteed this is somehow part of the biology, but there's a fundamental question when a scientist says we found, you know, DNA methylation or epigenetic mechanisms that are predicting something, I hold that as like an empirical commitment, but it might not turn out to be the case. So I think there's like at a very basic level, we're implicitly, even when I say I found an RNA marker, I found an FMR, I found a bold biomarker, I found a, you know, electrophysiological marker, I assume that that is actually true, and it might not be. So so I think from that sense, you know, there's very elementary causal commitments we're making when we in the science that we're doing. And so yeah,
that's almost a little bit even more worrying than we'd started with. It's not just that we have to worry about the causality, you know, within our neuroscience models that we're trying to build, but we have to worry about the causality even in our instrumentation and the way we're collecting the data, the way we're thinking about the process of actually measuring things in the world. Adeel, do you want to add anything briefly?
Yeah, definitely. So, I mean, I was a bit, laughing a bit when you asked about it, because that's what we do for our living. And so, if but Eric, you also bring. So you were talking about philosophy, and so that's so probably I will just go take a bit of direction in that. So, for me, it is all about consequences and the causes. So when you say correlation, so most of the people who are doing this, for example, in fMRI, 99% people, they are using correlations. And to me, correlations are just nothing but a consequence of something happening which is hidden or latent, and this is what we call the causes. And the causal relationship between hidden states that's causing that we see, that we observe as a time series, and then with time series we can just calculate anything we like, second order statistics like correlations, coherence, quasi-spectral densities, so on and so forth. So we can do so. These are all consequences. They don't tell you about anything about what actually is the mechanism, how they actually go about that. And for doing this, what we do is we build generative models. So we build generative models, which are the models that how the consequences get caused. And this means that when we build generative models, this means that we specify the states, the parameters, the models, and together we can interrogate them, and we can put different pieces together and try to think more mechanistically that how the things go about, that how the things happened in a way that they happened, and that's where for me that's a causal process. There is a so when we talk about, for example, we work with dynamic causal model, and sometimes it also creates a bit of confusion where the cause, when we use in DCM, we actually mean the causes that we are trying to infer from the consequences, and this is what we mean by that. Anyway, so for me, the whole thing is very clear. Is about when you are using functional connectivity, you're working with consequences. They're not going to tell you, you're working with the data observations. They are not going to tell you anything about that what actually happened inside the brain, which is hidden and latent. And in order to know how the correlations came about, we have to have a generative model, which we usually write down as a state space model and so on. Probably I will unpack this later on, but this is how I see the whole story within neuroimaging. Is I started with causes and then consequences, which is a bit more a philosophical take, but then, in terms of very narrowed down functional MRI or EEG, MEG space, I see these things from this lens.
So I think we'll come back to this, I'm sure. But the thing that's interesting in what you're saying, so we've been talking a lot about models during the Neuromatch Academy so far. And a generative model is a nice thing for us as a scientist, as a tool, because we actually can write down what the true causality is, right? And it seems like that's actually the easy part of the problem in some sense. If I want to write down a hypothesis about what causes what, I can simulate it in a way where I actually know that causality. And so part of what we struggle with is, I guess, figuring out whether that particular generative model is actually a true model in the world. But let's go to, we now have a good number of questions that have come in. And I think it would be great to start with a really basic one that should help us sort of clean the ground. So this is a question from Yong Cheng, who's asking, what's the relationship between correlation, independence, and causality? And he's asking basically, you're in a situation where we know that when you change one thing, it changes something else. That seems to be true of both correlation and causation. And so after thinking through this, and I guess this means that Conrad, your tutorial, he says correlation is causality. So I don't know. what is the relationship between correlation, notions of independence, and causality in a formal sense, so that we can just have a clear definition to begin with?
Manjari, do you want to give the definitions, or like, or what's the? I mean, like, let's briefly talk through the content of the tutorials. No, like when it comes to this, what we've seen is that if we have very small systems, then in many cases correlation is causation. What we've seen in the tutorials that if we have larger systems, then correlation and causation are often very, very different. So let's maybe structure that a little bit. And of course, Manjari is the card-carrying statistician here. But basically, let's say we have two variables that are correlated. Why could they be correlated? Well, there's multiple reasons why they could be correlated. It could be that A affects B, or it could be that B affects A. It could be that there's C that affects A or B, or that A and B affect D, and we've only incorporated all those trials where D has a certain value. Okay, so here we have like 4 different scenarios of why they could be correlated. Now, only some of them are causative. And so in that sense, correlation isn't causation. And really, for me, the gold standard is always perturbation. Like if I have a system, the definition of causation is if I would maybe make A more active, and then I see that B becomes more active. That is, as we saw in like the early parts of the tutorial, like a good way of defining causality here. And so without interfering with the system, unless it's fully observed, we cannot get at what causality is. So in that sense, I think it's easy to kind of get sidetracked like that correlation needs to imply causation. In big systems, the bulk of activity is like generated by itself.
Manjari, do you want to add something to sort of how we should formally think about this?
Yeah, so I'm not sure. I've been rereading the question from Wang Cheng, and I'm trying to get at, you know, what this question might be getting at a little bit. And one thing that comes to mind is that I think it's important to separate, you know, well, let's say there's just 2 variables, and we've made the claim that, you know, X and Y are associated or correlated with each other. What we're actually saying is that observing X is correlated with, tells us something about observing Y. But the way we talk about probabilistic dependence, usually in daily language, sometimes we might say, oh, you know, does X explain Y? We might talk about it more colloquially. And when Conrad and the tutorials today were talking about, you know, thinking about interventions, they're actually talking about manipulating it in a way. And even if you can do an intervention in real life, the question being asked is hypothetically, like, would you, if can you conceive of some kind of manipulation of one? And does that manipulation somehow change the distribution of X and now change the distribution of Y? And so when you're thinking about does changing X change Y, I think it's important to understand what we mean by changing. Do you mean changing in the way that X has been observed in the data already, or do you mean changing X in the sense of a hypothetical manipulation?
That seems really, like, I think to me that makes that much clearer. So I don't know, thinking back to my philosophical misspent youth, you know, when we were thinking about causality, it's not something that's ever actually related in the definitions, sort of philosophically, to observation, right? That isn't what we were looking for. Conditional dependence, right? The actual formal relationship where if I change something, I think about what will necessarily happen, or maybe probabilistically happen, but it's defined from that side. So I don't know, it seems to me like the simple analogy here is, it's not just whether we see a pattern But if we were to imagine ourselves having a lever where we could adjust variable X, it better be the case that when we pull and push that lever, whatever things are causally dependent on it shift in the way we expect. Is that a clear?
Yeah.
Yeah.
Okay.
If I just briefly add to this, in terms of like, not that I just wanted to have my turn, but it's just that because when you talk about the small systems and large systems, and you see for small systems, they say the correlation causation seems like similar. It's just because the correlations are the consequences, and it is when small systems are small, it is possible that we can track. It is possible for us to track what could have the mapping from causes to consequences, because there are finite number of things that one can think of. Yeah, that something happened. There are few events, and there are possibly a finite number of reasons that why it happened. And that's why you see, that there's once you can. So what I'm trying to say that if we can resolve the mapping, then they seem similar, and it's easier when it's a small system. But when the systems become bigger, the problem is ill-posed. The mapping from consequences back to the causes is an ill-posed, ill-conditioned, which means that we need to regularize and so on and so forth. There could be infinite number of causes, and there are many ways that you can think about that. And then because of the infinite number of hypotheses that we need to test to get to the observed or the consequence or the correlation, it means that we, the whole thing falls down. And you see, as the systems become bigger, correlation and causations seems like there's no association between them. So that's basically a very, explanation that comes from a bit more philosophical angle as well.
So one of the things that we'll touch on in a minute, I think, is this question of how things, as Conrad mentioned, seem to get more complicated as we think of larger scales. But Conrad would like to answer a question here by Xiaoqi Li on inferring causality. So Conrad, since you wanted to answer this question, I'll let you give an answer. I assumed you were motivated.
Great. I think this is a very, very important question. So let me read the question. The question is, can we infer real causality in the brain by simulating recurrent neural network or other realistic neural models? If they successfully replicate the data, can we then conclude that the model is right? And this is something that I think we will even have some disagreement about panelists. So there are two different intellectual traditions in this space of causality in networks. There is one that comes from the statistical field that's called causal inference, that in today's tutorial was largely following that first set. What would that field say about it? It would say, under almost all circumstances that we have in neuroscience, we're recording a very, very small number of dimensions. And you saw that we had a part of the tutorial where we only recorded a subset of the neurons. And you could alternatively record a projection of them, where you say, let's say, we only know average activity within each voxel or something like that. The first field, the causal inference field from statistics, would say, well, in that case, we're confounded, and we can use the omitted variable bias equation. We can therefore worry that things might not be possible. There is another field that kind of comes more from maybe an MRI data analysis background. And in that field, indeed, the idea is, let's start with models. Let's start with simple models, see if we can fit them, see which models we can prefer over which other models, and hope that over time we can make our models good and constrained enough that we can get at causality by fitting those models. Personally, I'm clearly in the first camp. So I feel that we will, when we record from, say, order 100 regions of interest, this is recording such a tiny part of the overall variance that we shouldn't be, that we shouldn't strongly expect that the output of an algorithm looking at these low dimensional projections would be would tell us so much about causality. And the reason for that is simple. And like if I say record from those 100 dimensions, and it looks like I often see like A happen and then I see B happen. Well, that could just as well happen through like a circuit of other variables or like common drivers between them. Like so in that sense, I'm strongly in the fast group, but I'd be very curious how Adeel feels about it, because what DCM does is it does fit models of progressive complexity to try and get at a notion of causality in many cases.
Yeah, definitely. Yes, so I think I largely agree with you if you what you said, and in a sense that what you were saying about like how there are two camps. Yeah. So I think what I would say, start by saying that you know there are two ways that we can, or possibly many more, but one is one is where we look at causality from as a time precedence. Yeah, that something happens first and it causes consequence. And the second one is about the physical, which is the control theoretic, which is the physical influence of one thing. And this is what I talk about when I talk about changes in causes, produces changes in consequences. And so for example, if I'm observing or estimating the activity at a certain network node that potentially provides information about its effects at remote nodes, however, physically acting upon or fixing, for example, this activity effectively removes any other physical influence this node receives. So this means that the inferences based on the effect of an intervention that we were previously talking about are somewhat different in nature from those based on purely observational effect. So there are two things. One are based on observational effect, where you just don't intervene. There's no intervention. While the other thing that I'm talking about, which probably which definitely links to how we see DCM, is on the causality in a controlled theoretic manner, where we have intervention, where we have exogenous inputs which are entering the system, and they are selectively perturbing certain neural system. For example, a visual stimuli coming into a visual cortex, perturbing that, and then there is a series of events which are happening, and so on and so forth. So I mean, I understand what Conrad you're saying is this, that the brain is a very high dimensional space, yeah, infinitely, yeah. While what we have is only very few observed variables, and from this we are trying to and which is basically p greater than n problem. Yeah, we have huge number of parameters, but we have very low number of very coarse observations, and it seems like it's an impossible problem to solve. And that's why I think only few of us are doing it. There are not many people. Like, there's another question which talks about why people are not doing causality, because it's different. It's a beast. So what I'm trying to say here anyway, so this is, so what I'm saying is this, that what we are doing in DCM is bottom-up approach. So one thing is that you look at data, and this is data driven, and you look at the data and you go back towards what could have caused the data. The other thing is this, that Conrad was saying DCM is doing, is this way of building generative models. We are starting from the bottom with some assumptions. If I just give an example, for example, those who use AEG, like in AEG, source reconstruction problem is supposed to be an impossible problem, because that's only possible if you first determine at what spatial and temporal scales that you want to work at. It could be at macro scale or a meso scale, what temporal scales you want to work at, what spatial scales you want to work at. And then you have to do some assumptions. For example, what we do in DCM is a Bayesian framework. We regularize a problem with a certain set of assumptions which can be challenged. for example. For example, the example of source reconstruction with AG is only possible to tackle that problem if you start with prior beliefs about what the problem look like, so that it is something which is tractable. So DCM framework is just about making making assumptions and developing models which are simplistic to make the problem tractable. Now the question is this: that are we getting the true causality? Is the way? Can we know? It's very difficult. But we hope that what happens is this: that once we have a certain, so the hope is this, that we can, because there's no guarantee. We don't know that certain brain regions, how they are causally connected in most cases. The hope is this: that we can reproduce and repeat those studies of different sample sizes, and probably at some point converge that, okay, these brain regions, this is how they are causally connected under a certain context dependent fashion. So that's the hope. But I can fully, I know I have to speak a lot, but what I'm trying to say is that I'm very much aware that there's another possibility that which comes from the idea that it's basically an impossible problem which is untractable or incomputable.
Well, so I think this brings us to a really important set of questions that were touched on in the tutorial, and Dibing Zheng has also asked about this. So we could make a choice, which is that for large-scale brain functional network analyses, we should only do truly causal analyses and truly causal experiments. And if that's true, if it's really easy to go wrong by using correlational analyses, then why are people still using correlational analyses, and why haven't we all switched to causal analyses? And then I think related to that is a question of what is a real causal analysis, because you're claiming something that is more like a set of hypotheses about a causal structure, and whether those hypotheses, generative models of causal structure, seem to be reflected in the data. So it's some kind of model selection, which we've talked about in NMA, to guess what the most likely causal structure would be, without actually having done something that I would think is really a causal analysis. So Manjari, do you want to talk a little bit about, how can we use correlational analyses, and what's the relationship between the kind of sort of causal hypothesis that's used in DCM and real causal analyses?
Yeah. So I really agree with your framing of this as model selection. So coming back to the original question that we were asking, the question was that, if we were to simulate a recurrent neural network and that somehow successfully replicated some experimental results, does this mean we're doing a causal analysis? And I would describe what is described in this question as actually checking, goodness of fit of like a particular model with the data. So this is pure statistics. Now it could well be that the model you're considering is the true model. So what you've done a check, and if the result had been false, you would have falsified your hypothesis. But if the result is true, all you've confirmed is that you have a model that is consistent with the data, and it's potentially one of many possible models. And I think that's where the two camps that Conrad pointed out, like the first camp, we want true causal identifiability across all possible causal models that would be consistent with the data. But that's a very high bar to set. And sometimes we might pragmatically say, well, we have a bunch of models available to us, and we'd at least like to do a good job of choosing between them and finding the ones that are most consistent with the data. And I think that's still the second thing, the second approach is still an important thing to do. And it could well be that you're able to distinguish between a few distinct kinds of causal models. You just haven't completely, eliminated the possibility that there's other competing causal models that you haven't even considered.
So that's an interesting question. So normally when we think of model comparison, you know, it's one thing to stack two models against each other and see that one is really dominating and then declare victory and go home. It's a sort of second thing to take a large family of models and really actually try to ask in this whole, can we exclude huge portions of this family? And I'm wondering, is there a way we can think about, you know, how powerful something like DCM or Granger causality analyses are in eliminating some of the sort of sets of that large model space? How much can we reduce it? Is it at least more powerful for us than just doing, you know, correlation analyses?
Yeah, I actually want to clarify that, you know, when and I think this is probably true of Conrad too, when I talk about correlations or associations, I include a very large family of associational measures. And so for me, Granger causality is really more like Granger dependency. So because what you're checking is whether, you know, the past of some variables or a certain class of variables are associated with or dependent with the future of other variables compared to if you were to switch them around. And so typical associations are maybe you treat them symmetrically, and when you look at measures like Granger causality, you're now introducing an element of asymmetry. And what you're basically doing there is that you're ensuring that you can eliminate, like what's really useful is that you're telling where correlation isn't. Like if things are not associated, if two variables are not Granger predictable, then it means that then you've learned something new. So I do think like Granger causality, especially when you're studying physical processes and things that change over time, it gives you more information than just a plain association if you're able to estimate it well from the data. So you are getting, in that sense, the more you're eliminating possibilities, you are getting closer to causality in a certain sense. So it's not irrelevant to getting at the final causal questions. It's just, you know, you've just found another, if something is Granger cause, if X Granger causes Y, you've basically established an asymmetric association, in my view.
That, yeah, that I think is really nicely stated. Conrad wants to say something.
I was just going to add one thing. A lot of people add something implicit there that I think is a big confusion, which is that people believe that if we take one of these correlational methods, like Granger causality, and we produce a lot of data, let's say we get like 10,000 subjects or something, that then the problems go away. But that is not the case. And like that is the omitted variable bias equation. So the bias that is introduced by that. So that basically, it's not the case that just getting more data makes the problem go away. At some level, for a lot of systems, unless we can really record all aspects of that, we might only be able to get it at positive causality by really perturbing the system. And I think that that's just like an unfortunate fact of trying to figure out how the world works.
Yeah, so I really want to iterate. Yeah, sorry.
No, go ahead.
Yeah, please go.
Reiterate what Conrad said there, which is that, you know, 10 years ago, there was an article in Wired that said, you know, with big data, the moment we have lots of data, we don't need to do experiments anymore. There have been articles written in the past that said we don't need RCTs anymore. We can just collect lots of data, lots of observations, and we'll know how things work. And so I think part of it is that no matter how much data you have, like so long as you're trying to understand mechanisms in the world, like that's the act of doing science. It's like saying you can't get knowledge for nothing. So you know, there's no way that associations will ever be causation in that sense. But I think it is important that, you know, coming back to like the small systems example, for example, if you had just let's say you had only two variables in the world, there were no hidden confounders, then it's very clear that a pure association would not tell you anything about causation, whereas directed measures of association would eliminate possibilities and tell you something. So, you know, I think it's more about, are we moving towards the goal, or are we at the goal? And I think that's the
That's well
Like
said.
you
So
do
when
need
you
at
said
some level
about
experiments to get to, you know, eliminating all possibilities. So
when you said about what you said about big data was interesting, because you know, when people say that we have huge amount of data, we don't need to do anything anymore. So this, like, you know, the idea that is data fitting what is data interpretation, and big data and tiny ideas. I mean, if you have big data but you have no ideas, it's not going to take you anywhere. I mean, I would rather prefer big ideas and tiny data. But if you have big ideas and big data, that's awesome. But you know, people, it seems like, you know, it seems like, you know, if you collect their, like, people who are going out and collecting 100,000 samples and all that, which is great. I mean, which is great, but it has to be backed up with you know, with ideas. What actually you want to do with that data? Data is itself, it doesn't contain the answers. It is only a way that you can use it and you know, and test your big ideas. So that's one thing. Another thing was about when you were saying about model selection, and Manjari talked about goodness of fit. I just wanted to just say at this that when you, what we are after is a simplest model, which is the best explanation of data. So it's not only about the accuracy, but it's also about the simplicity. And that's what we have when we have the, for example, we have free energies. Like in DCM, we optimize what we call free energies. Free energy is just nothing but a compromise of a trade-off between accuracy and then complexity. A model which is highly complex will, you know, will be probably very accurate, but it will be penalized for having lots of parameters and probably doing overfitting. So the idea is this, that the model selection based on this trade-off, which is, you know, accuracy and complexity, and that would be, you know, what we are after. A model which is simple enough, but it's the best explanation for the data.
Yeah, I think here one of the things that I'm learning is that a way I could think about the space of this problem is that there's the gold standard, which would be doing interventions and our, you know, randomized controlled trials, right? Then there's, let's say on the other end, we just do a giant GLM or LM, or even worse, a deep net on a huge data set. I know everybody loves deep nets, but it just pushes the problem somewhere else. And we come up with some amazing sort of inference that we can see something in the data, but it doesn't really help us very much in understanding. And one of the compromises it sounds like we could think about in the middle is using this sort of formal process of hypothesizing causal structures and looking at the possible outcomes we would expect to see. And maybe there's a number of ways, DCM being an example, where we can try to sort of at least get us into a space where we think we have a model that's both heuristically useful, something we can use to talk to each other about and seems like a good explanation of the data, that has an underlying causal structure, a real underlying causal structure within the model, that at least is compatible with what we've observed. And that's a useful maybe middle ground. But let's try to get to a couple of questions here that are, I think, core to the sort of basic question. So Leandro Schultz and Wei Zhang both are interested in sort of how we think about doing an interventional sort of model analysis where we have something like optogenetics that we're using. And we just would really like to know if I can't actually control something, one of these latent variables I think is causing it, I'm only capable of observing it. Like, how do I think about error and bias? And specifically here, I think maybe Conrad could start us off by sort of setting up the problem in the space the way it was set up in the tutorial, the way the students are thinking about, and then we can discuss.
Great. So both questions in a way relate to instrumental variables, where you could say if for every neuron there was a driver that drives those neurons, and we can fully observe that driver, and that driver is private to the neuron, then we can get at causality. And it's a typical case. We basically put in some prior knowledge, which there's these variables, and we know ex ante that these variables only affect the neuron itself and nothing else. In that case, some part of the activity of the neuron relates to that driver, and some part basically relates to the network activity. The parts that relate to the driver are by definition not causally affected by anything else. And therefore we can directly read out causality because effectively it's as if the world runs an intervention for us. So that's why it works very well. But let me also tie to the other side where the question was about optogenetics. So is optogenetics a direct readout? If your question is what's the influence of light on behavior, then yes. Like a lot of optogenetic studies, you stimulate the mouse using optogenetics, you see how that changes behavior. So yes, they are perfect if it's randomized stimulation. It's a perfect way of establishing that the light causally affects, say, the choice of a mouse or something like that. However, that's not how we interpret. Like what we say is we optogenetically, we put light into some area and therefore the area drives the behavior. Now that is much, much more complicated, because like the light affects countless neurons in that area, some excitatory, some inhibitory. In fact, it's a not so local signal. It affects neurons in large volumes of the brain. All of a sudden we are again in a causal situation, because if we want to know which neuron did it, well, we put in the light and the light affects all neurons at the same time. Therefore, the light acts as a confounder for all those different neurons. And therefore, it's very hard to know what the real causal chain is. And ultimately, the question we want to ask with optogenetics isn't, no one cares what light does. What we care about is what a given set of neurons do. And in the causal problem always comes back through the back door in that case.
So I don't know whether somebody has a specific thing to add to that, but maybe there's a related question here, actually. It's implied by what Conrad just said about optogenetics. So this is a question from Jihan Hachi. To what extent can we infer single neuron function from a population, given that most cells in a system are usually unobserved? Or is it better to rely on the ensemble or the population? So I guess, and now the last part is, can you ever have a large enough sample to approximate causality? And I'm not quite sure how I can sort of completely connect the first part and the second part. But I think what Conrad was just talking about to me is a very interesting problem, right? So in fMRI or EEG, we're measuring really gross signals. We're averaging over hundreds of thousands of neurons. And we want to make some causal claims. And maybe in what seems more precise state of optogenetics, we might be activating tens of thousands of neurons, or at least, many, many thousands. And now we want to think about this sort of causal statement. Is it something that we can attribute to a neuron, to the region, or as Conrad said, to the light itself, for example? Manjari, do you want to weigh in?
Yeah. So I think I think there's a fundamental issue. I was having a conversation with a philosopher of science recently, or on Twitter, and he asked this question. Why do so many papers that do experiments talk about like causal this and causal that? They're already doing an experiment. Like, why does the analysis have to be causal? And my response there was that the status, and this also is very intuitive to the statistician in me. So statisticians always make this distinction between the true unknown estimate and the estimate. We always put hats on our estimates to distinguish between our estimates from the population. And I think the similar analogy holds true for interventions. Like, there's always the perfect ideal experiment in our minds, and then there's the experiments we can do. And understanding the discrepancy between them is part of the modeling process. So even if you're doing experiments, you need to have a causal model for what the intervention does. And if that actually helps you get at the causal quantity of interest. So what is your causal estimate? What is your experiment? What is your model of the experiment? And does that get at your causal question? And I think that's where this discrepancy of, does optogenetics work? Does brain stimulation work? Like, all of these questions about experiments, they're often about, is the experiment adequate to get at the causal effect of interest or not? And in that sense, you might need to incorporate the non-idealities of the experiment into the process.
I know that helps me. I guess I want to ask one question because I think it's become another trend in neuroscience, and it relates back to the sort of two camps that Conrad we were talking about earlier. So in the way you've just described things, I can imagine a large number of papers that have been produced since optogenetics became really important for us as a new sort of causal intervention technique. And those papers do something like the following. There's a circuit between the mouse's nose and the mouse's paws somehow. And that circuit is going to be stimulated by, I apply an odor in the nose and the mouse does a behavior. And now I'm going to go and I'm going to take one tiny part of the entire brain, some particular nucleus somewhere, and I'm going to optogenetically perturb it. And then I'm going to say, I have found the circuit for X. I know exactly the causal structure from odor to paw movements, because in this sort of one-dimensional experiment, I have perturbed one aspect of I don't even want to think about the dimensionality. How do we even talk about that as neuroscientists? So what you were sort of saying to us here is we have to really think about this, like, what is our question? How close is our experiment to actually doing what we want to be doing? And it seems to me like the claims that I'm kind of making are, when a neuroscientist says, I've really done it, what they think is, wow, I set up an experiment that was my ideal experiment in my head. I really actually checked all of the possible perturbations for the entire brain. It turns out that this really informed me about the circuit. And so I worry a little bit, I worry about this kind of language, which has been absolutely pervasive for the last 10 years. Is this something we should be worried about?
I definitely think so. This is one of the things that makes me excited about causal inference, because they've thought so carefully for the last 30 years about modeling the like understanding the non-idealities of an experimental intervention. And in many cases, it turns out a decent enough experiment can go a very long way, especially if you combine that experiment with a clever, well-designed analysis, as opposed to you can do the naive, let's find an association between the experimental manipulation and the outcome of interest, or you can also design a more sophisticated analysis that takes into account the non-idealities and does something to address them, and in which case you get even closer. So I think that's it's worrisome, but also it turns out if you're willing to accept the non-idealities, you can do much more, you can do better than if you don't do that. So this reminds me a lot about what happened to, we take randomized clinical trials for granted, but when they were first proposed, people weren't sure. Is randomization the answer? How do we know that randomization works, right? And so first they did randomized trials, but there were no placebos. So they realized that they couldn't attribute everything to just because you did a intervention, you couldn't attribute the entire effect to the intervention only, because guess what? When you randomly assign somebody to get treated versus doing nothing, you're doing lots of other things along the way. By assigning them to get treated, they're going to a hospital, they're talking to a doctor, they're doing so like even randomization had off-target effects until we designed a placebo, like it was not controlled. And even after we did, RCTs, we didn't do any blinding. So it turns out that treatment for one individual affected treatment to another individual. And so I think some clever doctor in the 70s or 80s, I was remembering, I read this New England Journal of Medicine or some big, medical journal paper where this doctor got additional information, like to verify whether doctors knew who was getting what treatment or not. Because if they knew what treatment patients were getting, then they might subjectively report somebody doing better than they actually were. So they actually did questionnaires about how do you think, do you think this patient actually got the treatment or the placebo? So they collected lots of information like this and actually verified whether the outcomes were predictable, how often the doctors got it right versus wrong. And then it became clear that actually, doctors are able to tell who got something versus not because of side effects, because of whatnot. So that's when the double blinding became necessary. So if you look at the history of RCTs, people, it took them some time to realize, how to make that a better intervention, and or if they couldn't, what data should you collect so that you could, what additional information could you capture to account for the non-ideality of it? And so I think it's the same thing for us. This is like the doing the science part of science, like discovering the
it's more like
non-idealities.
evolution. Is the evolution of techniques and then methods that takes us, hopefully in the right direction. I mean, with optogenetics, I think that community has done a very good job of marketing themselves. So they can. So I'm very jealous of them because they can publish whatever they do in Nature, while us doing fMRI EEG, we can't, but anyway, I mean, what I could, there was a very nice paper about 3 years ago from a lab in Stanford, where they used Jinhuing Li's lab. Actually, what they did was to use because with fMRI, for example, just going back to a simple example, with fMRI, it's not possible to distinguish the cell types, but you could do it with optogenetics. Because you are more specific, you have more high resolution. So what they did was to combine optogenetics with fMRI, and that was pretty exciting. But for some reason, I haven't seen more work coming from that lab, and it's about 3 years ago. I thought that's very powerful, because then you can actually get the ground truth, because there you are actually very close to the reality. With fMRI, you are very, very far, and also you don't have the specificity. But if you combine these two, then you are really into the game. Or what Conrad was saying initially about TMS. I'm very excited about TMS because it allows you to perturb certain brain, areas, and then you can look at the causal influence. So I think optogenetics is always like, very, very nice tool. But, as you said, there's a lot going on there, which makes you cringe sometimes.
Well, I think what you said was really, the way I perceive it, it was very well marketed as the answer. But of course, as we've emphasized throughout the Neuromatch Academy, all of the kinds of things that we're teaching you, they're tools, and they have to be used appropriately by you as a scientist. You're somebody.
Yeah, and always we should believe in science, that the things which are sound, and they will eventually, stand out. So we don't need to, sometimes it can be a bit frustrating that one thing is hot than the other, while that things like these, it happens. But we should have the belief in the evolution, that the science will take us in the right direction, and things which are not stand to the test of time, they will fall away, and things which only, they will survive. So I'm pretty much hopeful about,
well, in the long run, that's going to work out, right?
Yeah.
The thing I worry about sometimes with these things is the efficiency of the process, because, as you know, we are, as scientists, we're also humans. We know we fail from the sort of kinds of base statistical inference problems that all humans have. We have to sort of overcome those by learning to do this kind of model selection and understanding causality and causal analyses and causal interventions. And that's something we train ourselves to do. But it's funny that even though we try to train ourselves to do these in sort of many aspects of our scientific lives, we still get caught up in other sort of simple human let's say biases. We fall in love with anything that's in science or nature because we have risen those up on a pedestal, put them on an ivory, column, and yet they're just another magazine, right? It's I don't know, I think of them as cosmopolitan because they're hot, they're trendy, everybody wants to see what the sexy thing on the cover is. But at the end of the day, what's going to really tell us about how things work is what still survives after many experiments, many replications, and many variations that will hopefully, help us to constrain our models and so on. We should close down because we're running out of time, but Conrad, I think he wants to say something. And I also wanted to ask specifically, Conrad, if there are any questions you see in the chat that we should give a 30-second answer to.
We can take them on your stars later. There's one thing that is really close to my heart. Like, we agree in a way that causal questions are the questions that are really central to what we mean when we say understand the brain. And we agree that we have worries about a lot of our techniques. I want to encourage the community to produce ground truth data on causality. It's absolutely doable. We can go into a system and perturb it at the level of single neurons, brain areas, microcircuits, many, many different levels. And whenever we try and make causal statements, we should try and, at least as a community, get meaningful ground truth data to test if the way we make inferences is actually right. And unfortunately, people jump the gun, they write lots of great papers, and then we find out that those great papers are like hugely misleading. So I want to encourage everyone to produce ground truth data to get towards causality.
That's a fantastic statement to close on. And I just want to highlight something that came up in the chat. So Leandro Schultz says to us, I plan to work with optogenetics and zebrafish, and I'm definitely going to be careful with my statements. So I think we've been successful today. And I look forward to the results that you'll publish in hopefully a couple of years. It'll be exciting to read them. In the meantime, thank you for joining us today for this question and answer session. I'd like to thank the panelists. Thank you, Manjari. Thank you, Adeel, and thank you, Conrad. Look forward to talking to you all again sometime soon.
Thanks a lot, everyone. I really enjoyed it. It was fun. Thank you.
Thanks.